In eukaryotic somatic cells, cell division is regulated by regulatory molecules which are activated at various cell cycle checkpoints. Cell cycle checkpoints are critical points in the cell cycle that are regulated by different types of regulatory molecules that will be explained in more details later. There are four main checkpoints, one in G1, one in G2, and two in M phase. In healthy, properly functioning cells, failing to meet the criteria for a checkpoint will result in cell division stopping until the requirements can be met. The goal of cell cycle checkpoints is to prevent the cell from passing along damaged DNA, as well as to keep the cell from dividing uncontrollably. Following the order of the cell cycle, the first checkpoint is in G1. To pass this checkpoint, a cell must first be large enough to continue to S phase. Secondly, an adequate amount of nutrients must be present in the cell's surroundings. Thirdly, there must be growth factors present. Growth factors are small proteins that come from other cells. They are found in blood serum and act as one of the catalysts for cell division. These growth factors are called social signals and are a part of social control. A system that ensures cell division is what's best for the entirety of the organism. And lastly, the DNA must be undamaged. Damage in a cell's DNA at the G1 checkpoint will put cell division on pause or trigger apoptosis, an organized death of the cell. Let's look into some of the regulatory molecules involved in the G1 checkpoint. P53 protein, on the left, acts as a braking mechanism in case of DNA damage and is responsible for the resulting pause or apoptosis previously mentioned. Other regulatory molecules at work in the G1 phase are E2F, RB protein, CDK, and cyclin. Here's how they all work together. When growth factors arrive in the cell in the G1 phase, E2F is produced. But because E2F is not needed until S phase, RB protein binds to E2F, rendering it inactive. When cyclin CDK dimers form, they bind to the RB-E2F complexes, phosphorylating the RB protein. When the RB protein is phosphorylated, it detaches from the E2F, allowing it to produce S-phase proteins, moving into DNA replication. Passing the G1 checkpoint is essentially the point of no return for somatic cells in cell division. If a cell passes this checkpoint, it will go on to complete the cell cycle, so long as there are no problems that occur later. The next checkpoint comes after DNA replication in S phase into the G2 phase. As in the G1 checkpoint, cell size, condition of DNA, and signals from neighbor cells are accounted for in these checkpoint criteria. In addition to these requirements, at this point in the cell cycle, chromosomes must be properly replicated. If not, then the inhibitory phosphate will not detach from the CDK subunit of the mitosis promoting factor, or MPF. The final aspect of the G2 checkpoint is the activation of MPF, since it is crucial in bringing the cell into M phase. This brings us to the last checkpoints, both in the M phase. The first mitotic checkpoint monitors the cell's progress from metaphase to anaphase. The criteria for this checkpoint is that the connectitores are properly connected to the spindle apparatus in order to ensure that the sister chromatids will be properly separated in anaphase. The second mitotic checkpoint lies between anaphase and telophase. Once chromosomes have been separated, enzymes that degrade the cyclin subunit of MPF are activated. Once MPF levels decrease, the cell is free to move into telophase. Now that we've covered how cell cycle checkpoints work, let's talk about what happens when they don't. So, what happens when the cell cycle checkpoints fail? The answer is uncontrollable cell division, which can sometimes mean cancer. Cancer cells are often deficient in important regulatory molecules, such as the P53 protein that we discussed when covering the G1 checkpoint. In fact, it can be hypothesized that many kinds of cancer stem from issues in the G1 phase of cell division. One example of these G1 problems is in defunct RB protein. If the RB protein does not function properly, then E2F would be allowed to produce S-phase proteins from the start of G1, rapidly pushing the cell into the next phase of cell division. In summary, the cell cycle is regulated by checkpoints. G1 checks for adequate size, nutrients, growth factor, and intact DNA. G2 checks for adequate size, growth factor, intact DNA, replicated chromosomes, and present MPF. M phase checks for connected connectitores and MPF turned on. And finally, malfunctioning checkpoints can lead to uncontrolled cell division and possibly cancer.